Good evening, ladies and gentlemen. My name is John Hill, and I'm chairman of Ulster Beekeepers Association. And I'd like you to welcome to the Ulster Beekeepers Spring Webinars. This is a series of 6 webinars, which we're giving weekly from the 12th of January to the 18th of February, and will be delivered by a series of very eminent speakers. We normally hold a conference at this time of the year, but unfortunately the pandemic has put pay to that. However, we do hope that we will return with a conference at Greenmount College in early February 2022. This evening's lecture is being delivered by Dr. Jeff Pettis from the United States on the impact of pesticides on bees. Dr. Pettis has been a former research leader at the U.S. Department of Agriculture Bee Research Laboratory in Beltsville, Maryland, and is now an independent consultant. If you're not sure where Beltsville is, it is in fact about 25 kilometers north of Capitol Hill, which will in fact be the center of world attention tomorrow for the inauguration of Joe Biden. Dr. Pettis is focused on improving colony health by limiting impact of pests, diseases, and pesticides on honeybees. And his research has included areas like integrated pest management and the effect of pesticides and pathogens on queen health and longevity. Dr. Pettis has served on several international committees, including the World Organization of Animal Health, the OIE, and is currently the president of Apimondia, which is the International Federation of Beekeepers Associations. So I think we can rightly say that he's king of the world. More than 35 years experience in research, conducted in many countries. And so I think we're delighted that he's here to speak to us this evening. Now, I would like to say a few words just to say that we're delighted that Thorn are sponsoring this lecture, and we are delighted that they have helped us out with this. And indeed, you can see on your screen that there is, in fact, an advertisement for Thorns, and also we will be having a short video at the end of the lecture. Could I also say that you have a facility on your screen for questions and answers, and I would ask you please to use that facility to ask your questions and answers, but please do not use the chat facility. Now, ladies and gentlemen, without further ado, I will now hand you over to Dr. Jeff Pettis.
Thank you, John. It's a pleasure to be with you, and it's a pity that we can't be in person, but it's such that it is this year. So I'd like to also thank Thorn for sponsoring the lecture, and I'm live right now. I'll be live at the end of the presentation. We actually pre-recorded the presentation as a precaution against some technical difficulty. So it's a real pleasure to be with you. And when I looked at Thorn's advertisement there, I was reminded that we're 2 countries divided by a common language, is that right? But also that our beekeeping styles are quite different, because we tend to use only 1 or 2 bee styles in the beehives in the US, whereas you use many different types. And we could get into a nice discussion about the WBC versus the national versus the Langstroth, et cetera, et cetera. But what I'll be talking to you today about is pesticides and their effects on bees and other pollinators. So again, use the question and answer thing to start to record some questions that you may have, and I'll join you after the presentation, and pleasure to be with you. Thank you. Hi, I'm Dr. Jeff Pettis, and we're going to talk today about pesticides. I really get tired of talking about all the negative things in beekeeping, and of course pesticides is one of those things. So I don't want you to go away from this meeting or this talk thinking that we're all in dire trouble and that pesticides are going to kill all our bees. What I do hope to get across to you is that bees do face pesticides in their daily lives, and some of the issues around pesticide exposure and how we as beekeepers can help to improve that. So I'm going to share my screen and start to talk a bit about pesticides. But again, let's stay on the positive side about we all get to work with bees. And so let me share my screen. All right, so the role of pesticides in bee health. Again, the idea here is not to scare you into thinking that your hives are contaminated and that all our bees are dying because of pesticide exposure, but to give a kind of a balanced approach to some of the things that are happening with bees and pesticides and some of the things that we can do to counteract that. So let's go. Well, honeybee colonies are dying at what I would consider an elevated rate. They, in the past, we used to lose 5 to 10% of our colonies through the winter primarily. And now, at least in North America, we're losing up to 50% of our colonies on a yearly basis. So we lose some in the winter, and we certainly lose some in the summer. And there's a variety of reasons behind those losses. So it's not just pesticides, but in different parts of the world, people are losing honeybee colonies at different rates, but also all pollinators are threatened. And we understand that things like habitat loss and pesticide exposure can play a role in those losses. So if you ask a bunch of beekeepers or bee researchers, why do you think honeybee colonies are dying? They'll give you a variety of reasons that they think that's happening. And some of that may be depending on what they work on. If they happen to work on nutrition, they'll say, well, obviously nutrition is the most important thing in honeybee health. Or if they work on nosema or viruses, of course, then they're going to think that nosema and viruses are playing a key role in why bees are declining. So there's a variety of things that come into play in bee health. And if you ask researchers the same way, or beekeepers the same way, they'll say, well, it was poor queens this year or starvation this year. So we all have our own opinions. What I think is playing in, I used to call it the 3 Ps, but now I call it the 4 Ps, which is poor nutrition. Nutrition is fundamental to bee health, pesticide exposure, and then pathogens. I've now kind of expanded that to I call it the 4 Ps, 4 Ps of bee health. And that is pest, like small hive beetle, varroa, things like that, pathogens, such as nosema, American foul brood, viruses, and then pesticide exposure and poor nutrition. And I think the poor nutrition aspect gets underplayed a bit. We as beekeepers and even researchers downplay the role that nutrition plays to some degree. I'll show you an interaction study here in a bit about pollen exposure and nosema increases. But in general, nutrition, if we have good nutrition, if our bees are well fed with a diverse pollen, they're going to do much better in all situations. So nutrition is vital as a starting point. And I bring this up because in the US, at least in North America, we've switched from being primarily honey producers. Most of the commercial beekeepers now derive most of their income from pollination fees. So on average, about 75% of the funds that beekeepers make, commercial beekeepers make on their bees, comes from pollination fees. And this has been a change in the last 10 to 20 years. Why is that important? Because if you're on pollination contracts, you're in an agricultural setting, and those crops could be sprayed with fungicides or insecticides and things like that. So the move towards more pollination in the US has certainly led to an increasing exposure to those bees to pesticides. And I'll cover some of that. But again, let's don't underplay the role of nutrition in bee health. As beekeepers, you know that we face our bees face a variety of pests and pathogens. Nosema, gut parasite, viruses are often exacerbated by varroa interaction, we know that. And even things like American foul brood. I'll tell you a little story about American foul brood. So it's almost everywhere in the world, and bees come down with it at a certain rate, but it's usually under stress conditions. And I say that because I ran for a couple of years, I tried to run American foul brood experiments where I took clean packages of bees, built them up, and then artificially inoculated them with American foul brood spores in sugar syrup. And I fed them over a 1.5-month period. They would not come down with the American foul brood disease. It wasn't until I got the spores directly into the cells where the young larvae were developing that they became infected. So, and I tried this with sections of scale as well. The bees cleaned it out. They did just fine in the spring. The key is this was in the spring. So when I ran these experiments in the spring where the bees were expanding and they had adequate nutrition, they were able to fight off all the AFB spores that I was pouring into their colony. Now, you as a beekeeper would never do that. But I was disappointed that I couldn't artificially infect these experimental colonies. And I think it all had to do with nutrition and how fast they were building up. But regardless, bees face all these different pests and pathogens. And we want to discuss a bit more in detail the role that pesticides play in their health. And pesticides come from at least two sources. Beekeepers, we're using things like fluvalinate and amitraz, and even some of the softer things like thymol. They do have an effect in the colony. So those are chemicals that we add to the colony. But certainly from the environment, mosquito control programs and crop protection where beekeepers, where growers are protecting their crop, spraying a product, or seed treating corn and soybeans, things like that. That can have an effect. And we'll cover a bit of that today in this lecture. A little bit about pesticide classes, not the different types of pesticides that are out there, not so that you memorize this, but there's things like insecticides designed to kill insects. There's miticides designed to kill mites. Fungicides are meant to kill fungi and keep plants from wilting and dying for fungal exposure. And then herbicides killing weeds that may be in a crop, things like that. We know very little about the effect of herbicides on bees. I'll say that at the beginning. And then down there at the bottom, you see a list of organophosphates, pyrethroids, carbamates, and neonicotinoids. And neonicotinoids have received a lot of attention in the past few years because they're a newer class of chemical. They move systemically in the plant, and then you can find them in low amounts in nectar and pollen. In general, you'll find them more in the pollen than you will in the nectar. But again, they move systemically, and they're also the most widely used insecticides currently. So there's a reason for concern. But a lot of what you hear me talking about today is fungicides. So, because I think they've been neglected in the past. Just a few studies that have looked at neonic exposure and done some surveys for pesticide exposure in bees in general. One by Marie Chazant and others in France in 2006, and Mullen et al. In the US, both found over 60% of the pollen samples contain some level of neonicotinoids in the pollen. Now, they were found at very low levels, 1 to 6 parts per billion and 1 to over 1,000 parts per billion, which doesn't sound like very much, but these compounds are active at very low levels. The Pettis study you see there at the bottom, I'm going to go into detail about that, so I'll save that. But that's where we looked at interactions between some fungicide exposure and nosema levels. Again, the picture up there shows us that, remember, this is apistan strips going into colonies. We are not completely free when it comes to the way we manage our colonies as far as pesticide exposure. So this study I was mentioning, it was done with my good colleague, Dennis Van Ingelsdorp, University of Maryland. And we went around the US and we collected pollen that was coming in while bees were on different crops for pollination. So they were placed on blueberry and cranberry and watermelons, things like that, pumpkins. And we collected the pollen that was coming in while they were on the pollination contract. And then we fed that pollen to young bees and exposed them to nosema. And to determine what they were feeding on the crop, would there be an interaction with nosema, and would nosema levels be worse? So that's the gist of the experiment. And I'll take you through it a little bit if you'll bear with me. So the field component is, as I've just explained, we used pollen traps, pictured there at the top. We collected pollen on the crop from 7 different crops. We identified the pollen type. So we went in and looked at the different pollens that were collected to determine if they were from the crop or from nearby surrounding vegetation. And then we analyzed those pollen samples for pesticide residues. In the lab component, we fed newly emerged worker bees with the pollen from that crop or for some pollen that we had collected in the desert southwest of the US, which was a mixed pollen source, and we knew it didn't contain any pesticides because we had tested it. And then after 12 days, we detected those bees and simply counted Nosema spores. So I'm going to show you a bit of the data from that. First, just a map. The pumpkin over there in the orange is about where I live in Maryland. The almonds on the far left-hand side of your screen are in California. That's a huge crop. It uses about 80% of our commercial bees, that one crop alone, almond trees. They bloom in February and March. And then bees fan out all over the US. And we collected pollen while they were on almond, apples, cranberries, blueberries, watermelons, cucumbers, and pumpkins. So from these 7 crops, in some cases we got late and early cranberries, things like that, but we collected pollen from each of those, and then we analyzed that pollen. So the first thing you're going to see is the proportion of pollen that came from the specific crop. So if we look at this first bar, let's see if there's my cursor. We look at this first bar, it's almond pollen. Almost 100% of the pollen that was coming in on those pollen pellets was from almond. And if we look at the next bar, it's apples. And in apples, it was only about 75%. There's 75% in this bar. Look at these other bars. Blueberry, cranberry, and pumpkin, almost no detectable crop pollen. That was pretty shocking, actually. And we were a bit worried about showing this information to growers. But the reality is the bees were collecting pollen from those crops, and they were exchanging it. It was getting collected on their body hairs. They were moving from flower to flower. It's just that some of those crops don't produce as much pollen, and the bees didn't have excess to collect and bring back to the hive. The other thing is that, and you'll see that if you understand some about bee biology, things like bumblebees have a different way of visiting blueberry and cranberry flowers, and they sonicate the flowers, and they remove a lot more pollen. So while we were surprised to see these low amounts of crop pollen, it wasn't totally surprising. The other thing I'll say about that is those first 2 crops, almond and apple, are from the old world. So bees evolved in the old world in Europe and Asia and Africa, and they're used to these crops. Whereas apple, cranberry, things like that, they're more new world. And in fact, the bees don't, haven't co-evolved with those particular crops. So, but this was somewhat of a surprising result regardless. Now we're going to take this pollen and we're going to analyze it for pesticides. This slide was just to remind me of that. This is an almond flower in bloom, and the bees do collect a lot of almond pollen during the almond bloom. So they're very efficient at removing this pollen and bringing it back to the hive. Whereas in crops like blueberry and cranberry, they collect very little pollen, but they do collect enough that they can pollinate the crop. Yes, bumblebees are better at pollinating cranberry and blueberry. Okay, so the number of pesticides detected is over here on this side, just the physical number of pesticides. So this first bar is white, and you see that in apple, we got about 15 pesticides detected in apple pollen. And the next bar, the dark bar, we have about, the pesticide load is about 9 parts per billion. So this, I'm looking for my cursor, there it is. This 9 parts per billion is of total pesticides. It's kind of low. If you go over here to pumpkin, there's more parts per billion in pumpkin, fewer total pesticides, only about 7 pesticides, but higher levels. So that's what this thing is meant to show. Now, if you have 7 to 12 pesticides in a pollen sample, how do you know which pesticide is causing any problems, or if they're causing problems? Well, we don't know. What we did is we fed that mixture, that pollen that had all these different compounds in it, we fed it to individual bees, and then we challenged them with nosema, and then we ask by using it's called a relative risk analysis, and we say, if compound A was in this sample and this sample and this sample, did the nosema levels go up? So it's very similar to what we do in human health when we look at risk factors. And this idea was kind of brought to honeybee health by Dennis Van Inglethorpe, and I thank him for his contributions on this. And I've showed this as neonics have been getting all the attention in the media, that everything is neonics, bees are dying from neonic exposure. And what I think you'll see from this is that fungicides are also playing a role. So we know that pesticides are bad. Which ones are the worst? Well, it's a mixed bag, if you will. So this is the type of class of compound that number of chemicals present by class. So this first class, this is all the crops. Those bars represent the crops. And this is the fungicides that were present in the various crops. So, there, in this case, the white bar in apples, there was, five fungicides present in apples. And in this other one, there was 7 fungicides present on, I think that is pumpkin, the bar here. Whoop, I'm looking for my cursor. My cursor is slow to respond. There's my cursor. This is 7 fungicides in pumpkin, 5 fungicides in apple. The key is to look at the group. There's a lot of bars here, fairly high. If you look over here at neonics, there's almost none. There's almost no, we could detect almost no neonics in any of these, all these crops, they were undetectable. They could have been there, but at low level. It was only in apple did we detect any neonic exposure in what we did. Some other things, pyrethroids are very common, and they use usually as a spray, not as a seed insecticide, but there's a fair bit of pyrethroids. But fungicides were by far the most common exposure that we had. Now, how do we make sense of all these different fungicides and things? And that's what I was getting to with this relative risk analysis. This is from human health and epidemiology. And we know in human health that if you smoke, you have a higher risk of developing lung cancer. And of course, the tobacco industry, we know that the tobacco industry has told us repeatedly that smoking doesn't cause lung cancer. And it's true, it only increases the relative risk by 230 times that you'll develop lung cancer. So it's not a cause and effect. And in fact, in our study, this is not cause and effect. Smoking doesn't cause lung cancer. It increases your risk of lung cancer by a lot. The same for coronary disease. Coronary disease, you see you have a 3 to 4-fold increase in likelihood of developing coronary disease if you smoke. You'll see our figures for increased relative risk with honeybees. It's in the 2 to 3 range. That's still significant. Anything over 1, where you start getting an increased risk, it gives you an idea that something's going on, that there's a problem there. And what you have to do then is go into further detail and find out exactly what's going on, what's the cause and effect. But these are the numbers for the relative risk of nosema infection, how much the infection went up with selected pesticides. The first 3 there, chlorothalonil, pyrrhostrobin, and captan, are all fungicides. And you'll see the first 2, there was an increase in relative risk, whereas the captan, it had negligible effect, no effect. If you look down there, bifenthrin, fluvalinate, and imidacloprid, you'll notice that imidacloprid in this study with nosema, it didn't seem to have any effect. In other studies it has, but in this study it did not, with all the mixtures. So I use this slide and just point out that we had the highest level of exposure to pesticides were in fungicide class. And that because we have these mixtures, we can't point to any one thing as being problematic, but by using this relative risk analysis, at least we can begin to get ideas of where to do future studies. And certainly fungicides warrant some attention from us as far as bee health. So the take-home messages from this study was that bees are exposed to multiple pesticides in the environment, in crops. And that's kind of known, but we just showed conclusively that there's a lot of pesticide exposure out there. Also, that it also doesn't always come from the crop itself. It can come from plants nearby the crops, either through drift or accumulation in the soil. And this has been shown by other researchers, Purdue and other places, that surrounding fields, the natural vegetation can pick up a high level of pesticide load as well. And then fungicides in particular, we show interaction with fungicide exposure, and in this case, no SEMA interactions. I'm going to talk about one more study relative to pesticides, and that is what I call the Swedish study. It was done published in 2015, and the Swedish government wanted to know, simple question, should they allow seed-treated canola to be used in Sweden? That was the question. The companies had come to them and said, we'd like to seed-treat canola, and it had never been allowed in Sweden. So they did a 2-year study, and what they did is they took, in new areas, they planted seed-treated canola, and they planted nearby other plots that were unseed treated, so just untreated seed of canola. And they put honeybee colonies, bumblebee colonies, and they put nesting blocks for osmia and other solitary bees out to see what would happen to the pollinators in these different plots. And they were big plots, they were hectare plots. And they did it at 8 sites throughout Sweden. It was a huge study. It's not one that's likely to be replicated, but some of the findings are interesting. So, excuse me, with honeybees, they had no significant effects on honeybees. Bumblebees, there was impact on colony growth. And if you know bumblebees, they build up from a solitary queen in the spring. They build the nest all through the summer, get to 300 to 600 individuals. Then they start to produce reproductives, female queens to overwinter. And the number of female queens that's produced is really important for the population survival. So in this case, the bumblebee colonies did not build up as well, nor did they produce as many reproductives in the fall. So they were impacted. Solitary bees, very few osmia nested in nesting blocks, and there was very little seen of the osmia on the flowers in the seed-treated group. So there was effects with the bumblebees and solitary bees. Why not with honeybees? Well, I remember Bayer's, when this paper came out, it was in Science, Nature, it was in Nature. And Bayer's comment at the time, Swedish study clearly shows no effect of seed treatment on honeybee colonies. They didn't mention the other two. They didn't mention the solitary and the bumblebees, but they were pleased to see that there was no effect on honeybees. Well, guess what? Honeybee colonies have a buffering effect. And I worked with some colleagues in Switzerland and was lucky to be on this paper with, you'll recognize some of those names, that we published an opinion that the superorganism, which is the honeybee colony, has a resiliency built in. They have a buffer, if you will, and they can take a certain amount of impact without showing any effect. A honeybee colony has extra workers that they can sacrifice. So you can think about it in terms of just the whole colony, and you could lose, 10 up to 20% probably, maybe more, of the workforce and really not dramatically affect the colony survival. So that's, we think, is just this buffering effect of honeybee colonies, that they don't immediately show effects when something happens in the environment, whether it's cold weather and some die off, for that, or whether it's pesticide exposure, things like that. So again, honeybee colonies do have this buffering effect. Superorganism resilience, but I like buffering, or built-in buffer. And this is kind of pointed out by some of these little slides I'll show you now. Honeybee colonies have large colonies, 30,000 individuals or more. Bumblebees, as we already talked about, they start from a single queen in the spring and they build up and then produce reproductives that overwinter. And you think about a solitary bee like an osmia or any other solitary bee, of course, they're totally dependent on that one female to collect the pollen, store it, and lay an egg. And that's shown in this next slide. All these 3 bees are using pollen, but they're using it in different ways. Honeybees can store pollen and nectar for a long, long time in the hive, and they forage over a much greater distance than do the other 2, the bumblebees and the solitary bees. So honeybees forage 5 to up to 10 kilometers away from the hive. 5 kilometers probably better. Bumblebees, shorter flight range, but they do store nectar and pollen. And solitary bees, most of them go out, collect what we call a pollen ball, put that pollen ball in a tube or a nest, and lay a single egg on it. So they're provisioning the nest with pollen they've just recently collected. So there's really no storage long-term of the pollen. So this impacts how the bees might interact with the pollen. And honeybees are quite different and long-lived. So how does that affect what we do in terms of how we look at pesticide exposure in bees and how the regulatory agencies look at it? Well, if a company comes to, in our case, the Environmental Protection Agency or an agency in Europe, the EFSA, and wants to register a compound, they only look at the data from that one compound. Let's say imidacloprid, which is a neonic. So is imidacloprid exposure to honeybees or bumblebees, is that safe? They can't regulate the vast majority of things that beekeepers come in contact with or bees come in contact with, nor can they look at the interactions. And that's just a fact of life. We can't ask the registrants, the Bayers and Syngentas of the world, to test every possible combination. So what do? They get licensed, they get put out in the environment, and then we look for those interactions. It's actually up to researchers and others to report when there's adverse effects. So it's not the perfect system, but I pointed out because if you saw from this study in cranberries and watermelons and stuff, the bees are getting exposed to a wide variety of pesticides. And we're not going to change the regulation thing. We can tighten the rules. We can make it more difficult and require bigger studies, but we're not going to change the fact that they register one compound at a time, and yet the real world bees are exposed to a multitude of pesticides. So now I want to tell you a little happier story, one that I was asked to cover, and it's that bees have good defense systems. They, if you go into a hive and you're not careful, they'll tell you about it. They'll sting you, but they have a variety of defense mechanisms at their disposal. And in some of the work that Dennis and I were doing, we started noticing what we called entombed pollen. And this is pollen cells. There's pollen under that black capping, and that black capping is actually propolis. So they've taken propolis and they've covered up those pollen cells. And this is on the same comb. These pollen cells are not covered up. And what we found, we went in and dug out these pollen cells and dug out a nearby pollen cell, and we analyzed this normal pollen versus this entombed pollen, we called it. And we looked at what was different between those two. And there's ongoing work that hasn't really been realized yet about, how this pollen was breaking down and whatnot. But the one clear signal we got out of that was relative to fungicides in that pollen. So again, remember, what I'm going to show you is the analysis of this pollen cell versus another pollen cell. And we collected that from this comb and from several combs across different beekeeping operations. So it wasn't just a single point in time. We looked at multiple examples of this, and all the pollen comparisons come from the same combs. They're mixed by comb. So the control is that normal looking pollen. The figures here in the control is the normal looking pollen. This is entombed or propolized pollen. The four compounds that we found most often were fluvalinate, apistan, coumaphos, which is checkmate, chlorpyrifos, which is a common ag chemical, and chlorthalonil. And chlorthalonil is a fungicide. So in the control, they were present, but they were present at fairly low levels. So these are very, very low levels of the insecticides or fungicides. Here, they were at much higher levels, especially chlorthalonil. Chlorthalonil is way up there. Coumaphos and fluvalinate are elevated as well. And you say, well, how could that be when here's a cell over here that we sample from, and maybe we sample nearby. How could that wax differ that much from here to here? We actually don't think it did differ that much. What we think is that it was some kind of chemical reaction with the fungicide and the pollen and the decay of the pollen in this cell. First, the bees detected it. They didn't like it and they covered it up with propolis. That's the good side of the story. The bad side is that we think that then there was higher chlorthalonil levels, that pollen, when it was brought in, came from a crop or something that had high chlorthalonil, and then it pulled the coumaphos and fluvalinate out of the wax, because these we commonly find in wax. These are get stuck in wax and they persist for long periods of time. So it's the best explanation that we have is that these coumaphos and fluvalinate were pulled out of the wax and into the pollen. We don't know that. What we do see is that chlorthalonil went really high in the entombed pollen. So that gives us some hope. Bees detect this pollen. It's inferior in some ways. It has high chlorthalonil levels, maybe other pesticides, and the bees seal it up and don't want to use it. And I've seen this repeatedly. If you look closely in your hive, you'll see some entombed pollen. You can take a look at it. The bees generally don't try to consume it. There's a lot of follow-up studies I'd like to see done, but I haven't had time, and I'm not sure who might get time to look at what are some other effects of this pollen entombing behavior. You know, they'll entomb a dead mouse in the bottom of the hive. So they sense that dead mouse is a bad thing, they'll cover it up. Well, this is the same behavior. And now I'd like to switch gears. This is a defense behavior, I would call it. And I want to tell you a little story about another defensive behavior that was recently published by colleagues of mine, Doctors Heather Madela in the pink. You see Heather Madela in the pink, and Gard Otis he's from Ontario, Canada, and she's from Wesleyan College in the US. They worked with Vietnamese colleagues, and they looked at a defensive behavior that Apis cerana, the Asian honeybee, uses this defensive behavior against Asian hornets. Now, there are a variety of Asian hornets out there. And so not all Asian hornets are the same. There's one that's recently been introduced that is in northeastern Canada and parts of the US, very limited introduction. There's a different species that's been introduced into France and quickly spread through France and is hitting bordering countries. That's a different Asian hornet. And then this is a different species as well, I think, but or maybe this is Mandarina. But in any case, the Apis cerana bees, when they encounter these hornets, they have a unique defensive strategy, and I'll tell you a little bit about that. So when Gard and Heather were working with beekeepers in Vietnam, they noticed these spots all over the front of the hive. And they kept asking, what are those spots? What is that? And they said, well, that's a defense against the hornet. When the hornets come, the bees spot the front of the hive and they cover it up, and it helps keep the hornet away. And they're like, what? Yeah, no, when the hornets come to attack the hive, the bees quickly start to spot the front of the hive, and that spotting helps keep the hornets away. Well, the question was, well, what are they spotting it with? And they kept saying, manure, shit, cow shit, chicken shit, whatever. And Gard and them didn't believe it. So they set up an experiment, and this is a clean hive. So they cleaned the front of the hives in an apiary, and then they waited on the hornets to come and arrive and start harassing the colonies. And sure enough, as the hornets arrived to, specific colonies, the bees would go out and collect dung or feces and spot the front of the hive. Now, to me, it's a fascinating story, and you'll have to just Google it. And bees using manure as a defense mechanism. It's just come out in the past few months. And this is Apis cerana. It's not Apis mellifera, but it also gives us hope that bees are pretty creative when it comes to defense mechanisms. My next slide, I have a video, but I'm not sure it's going to work. So if it doesn't, maybe we can show it at the end. Again, the video was supposed to be inserted, just showing them actually collecting it. I'll go past that because I don't think it's going to work. And we'll move on to the fact that, just kind of a summary. So bees have a lot of problems and a lot of things that try to get into the hive and affect its health. And I think historically, our losses, especially through the winter, were only 5 or 10%. If you lived in a good area and the bees were doing well, then you could expect a 5 or 10% loss through the winter. When we got Varroa in North America, our losses doubled, at least doubled. So we went to about 20% loss. Since then, in the past 10, 15 years, we've seen almost a doubling of that, but at least another 10% loss. So we're running anywhere from, 25, 35, 45% loss throughout the whole year. And our bees just aren't doing as well as they have in the past. And there's a variety of reasons for that. But I think one of the main reasons is our increased use of pesticides. And that's also beekeeper use and agrochemical use. So I do think pesticides are playing a major role in pollinator health. And I've been talking mostly about honeybees, but in fact, all bees are threatened, all pollinators are threatened. And pesticides, I'm sure, are playing a role in that as well. So what are some things we can do? We're not going to change agriculture overnight, but when we have large monocultures, first of all, that's not providing the kind of nutrition that bees and other pollinators need. So we need to try to work towards diversifying agriculture, finding ways to plant hedgerows and alternative cropping systems, things that allow for a little more diversity in the ag system. We also, in our beekeeping and in agricultural setting, we can move towards a more integrated approach and only use pesticides as a last resort. When I talked about seed-treated canola or seed-treated corn, things like that, that's all front-loaded. That's all prophylactic. You're treating the crop against a pest before it's ever present. And that may not be necessary in all cases. The growers are using it like insurance, which I totally understand. They're using it like insurance, but maybe that 5 or 10% increase in yield that they'll get, over a few years' time based on using that product is not worth it. Maybe it's better to take our chances and only spot treat when things become problematic. That's one approach. The other is we can do things like just planting more diverse things in our own backyard. So summarizing this, remember that all pollinators are threatened. I mentioned, I work with honeybees and you enjoy honeybees. But when the United Nations looked at this, they came up with 2 factors that are probably the primary drivers of pollinator decline: land use changes or habitat destruction and pesticide use. Because if you think about solitary pollinators, up to 70% of those nest in the ground. So there's a huge proportion of solitary pollinators that nest in the ground, bees in particular. And when you disturb the environment, that disturbs their nesting site. The Swedish study showed that hive size can matter and bees have this natural buffer, but we don't want to rely on that. We don't want to rely on the fact that bees have this built-in buffer and can take a little more abuse. We'd like to see things improved for all bees, all pollinators. And again, it's not just one simple thing that bees face. We're trying to work to limit varroa impact, trying to increase nutrition, keep American foul brood down in our colonies. If you find it, you burn it, things like that. So there's no one simple answer, but I would point back to nutrition as being the foundation of good honeybee hive health. And that gets to the point of we just need to increase forage diversity in the environment, whether it's the natural environment or the ag environment. Look for ways to do that. And this last slide is supposed to kind of summarize that. These are active steps that we as local people can take, or on the national level. It can be mandated that certain things are done to increase and diversify agriculture. So I'm kind of encouraged by that. That kind of concludes the pesticide part. I'm going to finish with just a little bit more, one more little small study. And again, the idea here is not to make you think about all negative things, but it has to do with a researcher came to me when I was at University of Maryland, when I was at USDA in Beltsville, Maryland. He was a researcher working on climate change. And he asked, how would climate change affect bees? And we talked about some ideas, and he was working with plants and plants' response to rising CO2 levels. And the question was, what is an important plant that honeybees visit, that we could target to look at rising CO2 levels and the pollen quality? And again, I talked about it and I said, well, I think it's goldenrod. So we know from human nutrition that certain things in our diet aren't as nutritious as CO2 levels go up. It could be the same be true for pollinators. So that was the question, overall question. And we focused on Solidago, the genus Solidago, and goldenrod, because it's an important fall plant. Produces a lot of pollen. The bees switch and go in the temperate regions. They go from producing summer bees to winter bees. And a lot of that could be driven by goldenrod pollen. So we focused on Solidago, and I'll show you the citation from this. And it was published in, 2016. we did a 2-year study where we had a lab component and we had a historical component. And I'll show you those in just a minute. But will rising CO2 levels affect the protein quality in our target plant, which is Solidago, goldenrod? Excuse me. You're seeing a picture of the Smithsonian Institution in Washington, D.C. And in the herbarium, we have plant samples that have been dried and preserved for hundreds of years. And we went back into the herbarium. They let us go in. And from 1842, we have samples of goldenrod from 1842 to 2014. And we took very small amounts of pollen from those dried specimens and analyzed it, up to 350 plants. We analyzed it over a 172-year period. So that's quite a time span. And we're going to look at the protein content in that pollen. At the same time, over here, these are tunnels where we can monitor or change the CO2 level. So we have high CO2 going to less CO2, and we simply grow goldenrod in those different environments, and we look at the pollen quality in the high CO2 to low CO2. And we know how much the CO2 level has changed. We already knew that. So we know what levels to target here to mimic what we're seeing from the actual samples from the herbarium. So I'll show you a bit of data from that. So this is the top graph is really the one that's important. It's the estimated percent protein in the pollen. These are 1840 to 1860, around 1900. These are all our older samples, a lot of different older samples. And the pollen protein content was around 18%. And then you see it start to change and it starts to go down. And the recent pollen samples here are down around 12%, about 12% pollen from 18% historically higher levels. That's a 30% reduction. The bottom is nitrogen. So nitrogen went in the opposite direction. The nitrogen content went up. But the protein is what bees are used to build winter bees. So again, this is from the herbarium samples. When we did it in the greenhouse, we saw a similar effect. And I won't show you that just for time. You can go to the paper. We grew them at low CO2 levels. The protein level was high. As the CO2 level went up, the protein content went down. So it was very consistent, whether it was in the lab setting or whether it was historical. We saw about a 30% reduction, in this one crop of one plant, Solidago. What does that mean? Well, I don't know. I mean, the reviewers were saying, well, why didn't you feed some of this less nutritious pollen and do all these studies on bee health? We didn't have time. We wanted to get this information out. There's a lot of follow-up that could be done. But if you were eating something and it was 30% more nutritious, or you had 30% better pollen, you could expect you might do better overall. Whereas if you're eating a slightly reduced diet, same amount, but not getting the same protein, you might be doing more poorly. And that's what we argued in the paper. We also don't know anything about other plants. Could be happening in many other plants, could be happening in none of those other plants. But I think it's likely that overall, some of the plants in our environment are less, the pollen quality is changing, let's put it that way. So this warrants further investigation. So again, this was just to show you that there's a lot of complexity in bee health, and we continue to work on it and continue to be optimistic. I will say that I'm always optimistic, and that is because we get to work with one of the most wonderful organisms in the world. If you're a beekeeper or a bee researcher, you get to work with honeybees, and the complexity of their lifestyle and the things they do and the way they bring us joy is kind of unparalleled. So, I mean, other people keep chickens and do things and stuff, but there's nothing quite like beekeeping. So I thank you for your attention, and I'll try to answer some questions that we have. So thank you very much.
Right. Jeff, thank you very, very much indeed. That was fascinating, and I think gives us a great deal of things to think about as regards what we're doing to the environment. So I think as we're starting to get some questions coming in, if I could possibly start it off, ask you about integrated pest management. In as much as I tend to think that a great many of the crops in the United States get this, and in Europe as well, we get this sort of widespread use of fungicides and pesticides, and it's almost done as a matter of course. As you said, very little targeted application. And indeed, I think, is that not the way agriculture has really got to go? We've got to really look at it much more from a targeted point of view.
Thank you, John. John, I'm gonna have to interject, and while I think about that question, I think we're gonna play the short video.
Oh yes,
so that we appreciate our sponsors. So while I think about that question, let's play the short video thanking Thorne for their sponsorship, and then we'll get back to your question. So
Thank you, Jeff.
Right. Well, again, thank you to Thorne for their sponsorship of this lecture. And as I say, I think I possibly jumped the gun there slightly with the question. Certainly, I would recommend to anybody, if they get the opportunity to go to Thorne factory in Rugby in England, it is well worth it. It's very, very interesting. So, Jeff, we would come back possibly just to the question. I suppose I started one off there with integrated pest management, and see what your thoughts are on that.
So yeah, thank you, John. It's a good question. So I know more about the agriculture in North America, but I know for the major crops, we're doing an awful lot of seed treatment. So it's all prophylactic. They seed treat corn, canola, cotton, almost every crop as crop insurance, and whether the pest is present or not. And certainly some of the studies that have been done on soybeans and other things have shown that over certain years, you don't get an increase in yield. In other words, they're using them, but it may not actually pay off in the long run. And certainly from an environmental standpoint, we'd be better off to spot spray, or like you say, use integrated pest management, scout if the pest is present, then you go in and you do a spot treatment. You use a lot less chemical. I will, I worked with the Department of Ag, and I can recognize the importance of farmers and the tough job that they have. But I think we've moved too far in an over-reliance on pesticides, and that we're using them too much, whether the pest is present or not. It's just become too easy to use seed treatment. So yeah, we can use scouting methods and find out whether the problem is there, and treat only if necessary. And there's a huge organic movement where sometimes you take your losses, or you use organic methods, and you know, some years you have some failure, but it's worth it to produce a nice, viable, good alternative. So, and the same is in beekeeping. I know a lot of treatment-free beekeepers, and beekeepers are using. I use only organic acids. I manage about 70 colonies, and I use only formic and oxalic, and some stock that I think is semi-resistant to varroa. So there are certainly ways in agriculture and beekeeping to use less pesticides. So, yeah.
Okay. Right. Okay. The first question is from Susan. She said the neonics that are shown to be in the pollen, was that on spray crops or treated seed? Does neonic cause the same damage in treated seeds, and does it end up in the food chain of birds, etc.?
Yeah. So good question. The vast majority of neonic use is on seed treatment, but it also is used sprayed on crops. I know soybean aphid is one good example where they go in and they actually spray if the problem is there with a neonic. So I can't determine. In the studies that we did, we were collecting it on the pollen. And I will say that when we looked at pollen versus nectar, there's almost no neonics in the nectar coming out of plants. Not that it's not there, there's very little bit that comes into the nectar, but more when plants are treated systemically with neonics, it moves into the pollen before it does into the nectar, much more so. And does it get out into the environment and affect birds and other things? Yes. I mean, so it depends. So it's in the pollen. Later, it could be in the seeds that birds are feeding on, things like that. Yes. So there is some we'd call them downstream effects. Again, finding ways to use pesticides in a responsible manner would get away from this a bit, I would say.
Right. Okay. The next question is really about the almonds, and I think we're always really quite astounded by the almond industry, in which you end up with about 70 or 80% of the bee colonies in North America end up in California at the end of January to pollinate the almond crop. And I think, if I understand, it's approaching 1,000 square miles of almond orchards, I think.
So, and some recommend 2 hives to the acre. So we're almost at 2 million colonies, and we only manage 2.6 million colonies.
Yes.
So it's highly migratory.
I think, Mick was asking, what about the pesticides in almonds? Then it must be a huge problem then, potentially, with pesticides in the almond crop.
Yeah, pesticides and fungicides. And in that study that we did, we had bees collecting pollen on almonds, but we didn't get it done in time to include in the feeding studies and stuff. So a lot of people were like, wait a minute, you missed the most important crop in the US. So we didn't get the almond pollen analyzed for pesticide residues. That's why it doesn't show up in there. We did actually collect it. It just didn't end up in the study. We were there, and it was a logistics thing that happened. They do use a fair number of fungicides, more so than insecticides, on almonds. So almonds, bees have historically come out of almonds very strong. They get a lot of pollen and a lot of nectar, and they've come out in the spring very strong. But that's not always the case. Yeah.
Right. Okay. Then, whoops, hold on, just see. I think it was from Susan. She was sort of saying, very, very good, lovely talk, but asking really why fungicides have not gained the same public notoriety that neonics have, and indeed, is it affecting the bee populations?
Yeah. So I was reading some of the questions before, so I'm a little bit prepared for some of them. When they do the testing, when, let's say, our Environmental Protection Agency, and I've forgotten the agency in Great Britain, but when they ask a Bayer, let's say, to submit a study, they treat the bees, and then they treat the crop and see what happens in the crop. And with fungicides, when you directly apply them to bees and things, there's not much evidence of harm directly, where there is with insecticides. So like the things like neonics, they can be highly toxic directly to the bees. And I think the fungicide effects that we're seeing are secondary. They're interacting with other pathogens. They may be interacting with some of the pure insecticides and causing problems. So yeah, they have historically, the growers and the industry has thought that fungicides don't really affect bees that much. And I think a lot of research is showing that in fact they do, but some of this damage is indirect. And so the other reason neonics get all the attention, they're the most widely used class of insecticides now. And almost all the seed treatment involves neonics. So they're putting it on the seed, it moves up into the plant and gets into the flowers and protects the plant and the leaves during the growing season. But the bees are visiting those flowers and picking up bits of the neonics as well.
Right. So the next one is some area about entombing. I have to say, I hadn't heard much about the entombing. That was news to me. Very interesting. And then, well, Mary's asking, are associated entombed pollens with toxic pollens, like rhododendron?
Yeah, I read that question, and I'm like, I don't know. We need to find out. And in fact, right now we only can link it with chlorothalonil, one compound. I have to think that there's other things that happen maybe within pollen where it might change, and certainly toxic pollen, it could happen, but I don't know. I guess that would be something for future study.
Yeah.
Yeah.
I suppose that the ongoing thing is to know, how do the bees know that the compound is potentially toxic? So anyway, move on to Wally's. Understand honeybees have physiological and detoxification systems that work for them until they are overloaded. Would Dr. Pettis care to comment?
So yes, bees can detoxify certain compounds, and but because this is what we found out when we did the honeybee genome. If you take a stink bug, and you look at their defense mechanisms, they're feeding directly on the plant and on all, they get exposed to all kinds of things. They have a ton of detoxification enzymes and things in a stink bug. Honeybees feed on nectar and pollen, and we like to think of them as pure. So we were actually surprised when we sequenced the honeybee genome. They have very few detoxification mechanisms. They do have some, but like compared to a stink bug, or you think of a milkweed caterpillar that can detoxify milkweed plant and stuff, honeybees they're not defenseless, but they have a lower natural defense against some toxins because historically they fed on nectar and pollen, which is a pure product. So yeah.
Yeah. Well, I think Peter was really wondering why maybe almonds weren't included in your results.
Yeah. Again, we should repeat it and find out. I did. We don't know how much is being picked up in the almonds. We didn't get that analyzed in time because we'd already sent the samples off and we were doing some feeding studies when the almond pollen came in. I will say that I've done some work in almonds with spraying fungicides during the day or at night. And by simply changing when the fungicides are applied, if you spray it during the day when the bees are actively foraging, there's much more exposure. So we are trying to look at some simple mechanisms to reduce exposure. But unfortunately, I don't have the information on entombing or anything with the almond pollen. So yeah.
Right. Okay. The next question is where we got to. I think there was a question about the reintroduction of neonics in Britain, but I think it's passed on to that. So
it's still there, John.
The UK has approved use of neonic. The background to this is that it was sugar beet growers in England have been allowed to use neonics against to prevent a virus disease called yellow leaf virus, which is spread by aphids. And this goes against what the government kept telling us, that they were banning neonics, and suddenly they've approved it for use. They're doing it under what they call emergency legislation, but I think it's caused a bit of consternation is that they've been allowed, they've started to use it again. So
not knowing all the particulars, but I think you still have a ban on using canola. Is that right? Or
sorry, a ban on
the use of neonics in canola.
Yes, well, it has. Yes, I think all the main crops, all the main neonics have been banned in the European community for the last number of years.
Yeah.
They did allow them at one point, and they still, though they are still allowed to be used in greenhouses, but that but outside use is completely banned.
Okay, so I guess, I mean, that's good in a sense because those are the flowering things that bees visit more often. With sugar beets, I don't know the level of exposure, so I can't comment on it. But occasionally you will get a situation where, you know, if there's something that's really going to put the farmer out of business, they do apply for emergency use. And so, yeah, no, I'd like to see a world where we're using much, much less insecticide because we know there's two things: habitat destruction, turning places into cities and disrupting the ground and stuff like that, and pesticide use are probably the two biggest things that affect all pollinators. Mm-hmm. I can't directly comment on the sugar beet so much because
I think it's relevant to the transportation of bees. This migratory beekeeping that goes on for the almond industry. And it's being asked here, is the commercial transportation of bees a significant health factor in the US?
So that's a question that we've always gotten. And I will acknowledge that our beekeeping practices in the US are quite different than most other areas of the world, except perhaps Australia. We move a lot of bees in a lot of locations across the US. But we did this survey where we asked beekeepers about their practices, and for the 8 or 9 years that I was involved in those studies, we always ask them, are you migratory or are you stationary? And we'd ask them their total losses. So the simple question was, are you a migratory beekeeper or a stationary beekeeper? And in 8 out of the 9 years, the migratory and the stationary lost the same number of colonies. So 1 year, the migratory lost more than the stationary beekeepers. And we've done some work, and there is stress on bees. I mean, it wouldn't be stressful to move bees, but I don't know that it's the driving factor in bee losses. I don't know how much I said about that, but I did. The beekeepers are shifting. They're getting most of their money from pollination, so they move up to 3 to 5 times a year for pollination. And I think it's the movement, placement, and then the exposure to fungicides and insecticides on those crops that's kind of driving the majority of the losses. And yes, then there's stress from the moving itself. So they're not stress-free to move.
Right. Well, I think from Steve, the next question is really to make comments on the biological effect on honeybees from the beekeeper. In other words, we're down to the smaller units really, and what effect that, say, treatments were likely to have on the normal beekeeper and his colonies, and whether or not there's an effect on the queen spermatheca or resilience to deformed wing virus.
You know, with pesticide exposure and stuff.
That would be treatments, normal treatments, which would be well, in this country we don't have Coumaphos, right. But we do have Apivar, we have Apilifar, we have oxalic acid, and we have formic acid.
Yep. So there's something interesting when you treat with formic, and I don't know exactly what it is, but depending on a little bit on temperature and a little bit on humidity, sometimes we lose an excessive number of queens with formic treatment, not always. And so I don't think anyone's ever exactly figured out under the total number of circumstances. I mean, high temperatures above 38 or 40 C, you're going to get lost. There's too much of a flash evaporation of formic. With some of the other compounds, I did some work on fluvalinate and amitraz in queens and didn't see a lot of negative effects. We've looked at some ag chemicals and we've seen some loss of sperm viability in the spermatheca if the queens were exposed to some of the ag chemicals. So, and actually, Coumaphos is definitely a bad compound. It's bad for the environment. It's bad for beekeeping. We saw effects of Coumaphos treatment in the hives on queens as well. But I'm only using organic acids, and I think most of those, there's just going to be an immediate effect, if there's any effect at all, and it doesn't persist. That's the beauty. There's no residues from those compounds. I hope that kind of answered the question.
Right. Okay. I'll come on to think about recording with the at a later time. Well, it comes down to a question about Apomondia. I've lost it. Yeah. Please, Apomondia, look at the question of contaminated foundation, especially bright yellow foundation now on sale. Please refuse to even enter the highs when it's present. I wasn't aware of this from Judith.
Ooh,
I wasn't aware of this.
well, I don't know, and I haven't seen. There's no reason to add. First of all, if there's no reason to add a colorant to wax to make it more sellable or something. You know, I mean that. But I know of the problem in Europe where there was some wax adulteration a few years back, and that certainly is bad. Where they're adding paraffin wax to beeswax and making foundation out of it, and there's a problem there. Yes, if the wax that's used to make foundation has been used in a commercial setting, and a lot of beekeepers, there could be contamination of some of those chemicals in the foundation. There are ways to treat it with and try to get some of those compounds lower in the foundation. But the best way to know is there used to be a system in the U.S. where you could take your wax to the wax person. They would use your wax to make your own foundation. It also kept you free of any American foul brood spores and stuff. But I'm not aware of this bright colored foundation, and but there's no reason to color foundation. That seems counterintuitive to me.
Right. Okay. I think the next question from Mick is a little bit similar to that. It is with the fungicides and pesticides that might be present in wax when people reuse it. Then are the chemical levels building up? I think that's what he's asking about. Will that affect the fertility?
Yes. The short answer is yes. And so one way as a beekeeper to get around that is to, I try to go on about a 3 or 4 year rotation with it. If I have 10 frames in a box, I'll try to replace 3 frames every season. And over time, then I'm getting those older combs out and melting them down. And then you're getting new foundation in there. So yes, there is buildup in wax of most of these synthetic pesticides. Things like formic acid and oxalic, no. But yes, certainly with the hard chemicals, they do build up in wax.
All right. Okay. Get us a beekeeper, anonymous one, who's noticed sealed pollen cells, and would say the colony benefit from the regular comb movement rotation.
Yeah, and actually with entombed pollen, what I've actually done is just going, because they're usually there's not that many. You could either recycle the whole frame. I actually like to just dig them out and then and have them rebuild that area. Sometimes that works depending on your foundation though. And again, I tend to rotate, and hopefully each comb is not more than 3 to 5 years old. In Switzerland, I worked in Switzerland for a year, and they were adamant about replacing comb all the time. They were wanting that new foundation in there all the time. And of course, the bees do better with that, but it's a lot of work. If they're building wax, you're not getting as much honey. So it's kind of a trade-off there.
That's right. I would tend to think that's probably a thing in very recent years, especially it's spreading through Europe, is much more the idea. Instead of changing a third of your frames every year, you change 100%. Does seem to be an increasing procedure.
I saw that in Switzerland. They were changing,
Right, was to do with differences of pesticides and fungicides on different types of honeybees. So it's whether or not some strains are more resilient to pesticides or fungicides than others. I think it's
I did a little bit with fluvalinate and different races of honeybees and saw a little bit of difference. I didn't see a great deal of difference. What does differ greatly is like between bumblebees and osmia and honeybees. You can get sometimes it's that the osmia is always more susceptible, but other times maybe the honeybee is more susceptible. So there are differences between the bigger differences in bees, but there are subtle differences in the races of bees, like a Carniolan versus an Italian. But I don't know it for all compounds. So I don't think, well, that was lead to a question. Why don't we select for a pesticide tolerant bee? And I'm like, no.
Yeah. True.
Let's reduce pesticide use. Yeah.
Right. I think we're coming really probably quite close to our time here. And I think the next one was in relation to entombing. I think I think the indication of this is really do our bees say coming from different sources, and so some cells become more contaminated and others be less so, and therefore the bees are able to detect different cells. I think that's really what William is asking here.
That's what we assume, because that pollen was stored very close to each other, and we have to assume that maybe this pollen came from this plant, let's say blueberry, and that had been treated with the fungicide, and the other pollen was not as contaminated. But again, we don't know that, but that's an assumption that those particular cells had higher levels of the fungicide in them. Yep.
Right. So, well, it's against with your Appomondia hat on. It says, so why were so many honey samples rejected at the last Appomondia?
This is my Appomondia hat. I started when I moved to Appomondia, I started wearing that hat. It was the first year that we increased the testing level, and we're still working with that. I think part of it was a small percentage was actually not true good honey. Another percentage, the honey, when they ran it against the database, there weren't some of those honeys in the database. So when they used NMR or some of these more sophisticated techniques, they didn't match up and they looked fake when in fact they were real. So there's a big issue here. We want to increase the testing, but we have to be careful that we're not excluding some really good true honeys. So if you've got heather honey, you've got some unique honeys, get them in, send them in. There's protocols to get them into these testing laboratories so that your particular honey is represented in the database, and then it'll come out as true honey. So yeah, it was a bit of a issue, but it does highlight that some of the honeys did contain like some antibiotics and things like that, which should be forbidden. So it's worth it. But when I talk about honey fraud and stuff, it's this two-edged sword. You want people to be aware of it, but it also it's a danger of hurting your honey market. So know your local beekeeper and buy his or her honey.
Right. Okay. Well, I think we really got to make this really almost like the last question, Jeff. And it's from Una, and she says, "Is there any evidence of pesticides ending up in honey?"
So we do this food thing in the U.S. where we test store shelf food, and honey's been tested several times, and it comes out almost completely clean. The few things that they found in there are generally a little bit of fluvalinate, a very little bit of fluvalinate or something. So what they found more often has been some of the miticides that we've used. I don't know about the European testing, but again, when honey crosses borders, it should undergo the strict testing. And in general, honey is not as likely to pick up pesticides because it's water-based. And most chemicals are lipophilic and they move into wax. So that's a beautiful thing. That they move into wax, they stay in your foundation or in your hive, but they don't get into honey as much. And so yeah, we have to be careful, but luckily honey is still a very good pure product in most cases.
Well, Jeff, thank you very, very much indeed, both for the talk and for the perseverance of getting through all these questions. I would like to apologize to those who have asked questions here, but unfortunately we've really run out of time. And perhaps maybe you could hold those questions for another time.
Well, actually, John, if you will cut them and paste them and send them in an email to me, I'll do my best to answer them by email.
Okay.
I'd like to say,
see if we can do that. Right. Again, really, I would like to say is that we have 4 more webinars in this series coming up, and the next one is on Wednesday, the 27th of January, which will be on the landscape and bees, and that will be presented by Professor Jane Stout from Trinity College Dublin. Jane Stout is a very major really mover and shaker in the All Ireland Pollinator Plan, and certainly I think that you'll find that her talk will be extremely interesting. So, ladies and gentlemen, I'd like to thank, as I say, thank Jeff again for his contribution here this evening, and to thank you all for attending. And we look forward to seeing you, say next week to listen to Jane Stout, And then Jeff will be speaking again on the 18th of February on why queens are failing. So thank you very much indeed, and good night.